Hello, my name is Mariana Ferrarini. I'm from the INSA University of Lyon in France, and I'm here to present a comprehensive analysis of human SARS-CoV-2 infection and host virus interaction. This was a joint work from a collaborative international team coming from several institutions. This is the overview of our poster, and as all of you know, COVID-19 emerged in December of 2019 and has already infected more than 10 million people worldwide, accounting for over 500,000 deaths by June 29th. In this work, we present the analysis of the human transcriptomic response during SARS-CoV-2 infection, along with predictions of host proteins interacting with the viral RNA. In the first part, we used different methods in order to detect differentially expressed genes and isoforms. We performed several enrichment analyses, and we integrated our results with the Human Metabolic Network. Finally, we searched for differentially expressed transposable elements, or TEs, which are portions of DNA with the ability to move, and that have already been known to regulate gene expression. From the differential expression analysis, we tried to pinpoint genes specific to SARS-CoV-2 infection, and for this, we used a publicly available dataset encompassing data for 3 epithelial lung cell types, NHBE, A549, and CALO3, infected with SARS-CoV-2 along with other viruses. For example, we detected genes in the PI3K-AKT signaling pathway, and we found enriched functions related to immune system, phospholipid metabolism, stress response, among others. Moving forward, we also detected changes in isoform usage. And here, I want to highlight specifically the differential usage of interleukin-6 isoforms in both NHBE and A549 cells, linking this back to the immune system as previously mentioned. And maybe some of you are not familiar with, but transposable element, TE, transcriptional activation, has been frequently observed upon viral infection, and TEs have known to play a role in immune gene regulatory networks. Thus, we detected 16 families of transposable elements differentially expressed in SARS-CoV-2, some of which were already shown to participate in interferon response. But how does this work exactly? So TEs are able to activate the transcription of nearby genes. And when we looked further into the functional enrichment of these genes, we found enriched terms related to immune system, phospholipid metabolism, and more strikingly, progressive pulmonary function impairment. In this way, we were able to not only detect differentially expressed genes and isoforms, but also some of the regulatory factors playing a role in this infection response, as you can see in immunoregulation and phospholipid metabolism here. Moving on to the second part of this project, we predicted human RNA binding proteins, RBPs, that possibly interact with the viral RNA. This could in turn influence SARS-CoV-2 replication, transcription, or translation, or it could even hijack and dysregulate these proteins from the host machinery. So we looked for RBP conserved binding sites in SARS-CoV-2 genomes, and we gathered expression information for them. So first, we selected position weight matrices from the ATTRACT database, and we scanned the genome of SARS-CoV-2 in search of RBP binding sites. Then, we calculated the enrichment by shuffling the same genome sequence 1,000 times. The binding sites were found either in the positive stranded genome, including 5' UTR, 3' UTR, or intergenic regions, or in the negative stranded intermediates. Finally, we tested if these sites were conserved among around 25,000 SARS-CoV-2 genomes available from GISAID, and we validated 18 RBPs having enriched and conserved sites among these SARS-CoV-2 genomes. You can find more information of certain proteins and their expression in this table, but for instance, PABPC1 has already been experimentally shown to interact with the SARS-CoV-2 N protein. It was specifically upregulated in CALO3 and A549 SARS-CoV-2 infected cells, and it is expressed in lung samples and has conserved binding sites in 3' UTR intergenic regions and positive stranded genome. Overall, in this work, we try to pinpoint genes and proteins regulated specifically in the context of SARS-CoV-2 infections when compared to the other viruses tested, some of which have already been described to be important in the context of other viral infections. Many of the genes identified in this work are interesting and worthy of additional experiments to validate the analysis and predictions presented here. I would like to thank everyone involved in this project, along with our funding agencies, and more specifically to thank my co-first author Avantika Lal, who performed most of the RBP analysis in this work. I would also like to acknowledge that this collaborative team started in the context of the virtual biohackathon for COVID-19. Thank you all for your attention.